To our next question as it relates to this word sensitivity and specificity of our testing of the biomarkers. I know there's lab definitions for things, and then there's like general population definitions for things. So help us to understand what this means in a laboratory context, please.
Well, I think important to note is exactly what you just said. We get that question asked to us a lot. What's the sensitivity of the GI effects, or what's the specificity of your Nutraval? And I think there's a very important distinction to be made there when we talk about sensitivity and specificity. There's the clinical aspect of it, and then there's the actual laboratory analytical piece of it. So clinically, it really speaks to how good is this test at ruling a disease in or out, versus when you're talking about a laboratory kit for a specific biomarker. And so that comes to the whole concept of biomarker versus panel, Michael.
Yeah, absolutely. It's really important to make that distinction. I think a lot of times when we get questions about sensitivity and specificity, it's a question of, the GIFX dual profile, what's the sensitivity and specificity? When really, when you're talking about sensitivity and specificity, you're asking the question, how good is this single biomarker at ruling in or ruling out a clinical condition? So an example would be the C. difficile enzyme amino acid, the EIA for C. difficile. The question then becomes, how good is it at determining whether somebody has C. diff or not? And so what we do is we take patients who we know have C. diff infection, and then we compare the C. diff EIA test, and then we say, okay, 99% of the time when the patient has C. diff that we know of, it was positive, and then vice versa for specificity. When we take a group of people we know are negative for C. diff, this marker can predict how often, 99% of the time, that it's accurate in telling us that they don't have that condition. So the take home there is that sensitivity and specificity relates to how a biomarker can accurately predict whether or not somebody has a specific disease. When you extrapolate that to a big profile like the GIFX, where we're looking for maldigestion, we're looking for short-chain fatty acid production, we're looking for a lot of different things, then you can't get a overall sensitivity specificity, because the terminology is not really it doesn't align. Sensitivity specificity is, what disease are we talking about? What biomarker are we talking about?
Yeah, so I think when we talk about it, or when clinicians write in to ask us these questions, it's important to be very clear about the question you're asking, because we always come at it from that clinical perspective. Like Michael said, just the overall clinical perspective versus the analytical. So be very clear about those distinctions there.